so I will continue and let's get down to business. So Drosophila life cycle. Here you have a picture depicting the life cycle. So as I mentioned in the first slide, there is an egg in the beginning here. In this egg, these are the egg filaments in the anterior of the egg. So here is anterior, here is posterior. In this eggshell develops the embryo, takes, depending on the temperature, at 25, it's 1 day. Then a little larva hatches here. There are 3 larval stages, first stage, second, third, and you can see the size of this larva increases dramatically. So during these larval stages, the larva basically sits in the food and eats and eats and eats, and everything inside grows. Then at the end of the third larval stage, the larva pupates, crawls out of the food onto the side of the container in which it grows and pupates, and then within the pupa, the larva changes its shape dramatically from a little worm into an insect. So as I said, it takes about 10 days. Life cycle is fast. And if you want, of course, this is very nice if you want to proceed with your experiments, but if you are keeping thousands and thousands of different fly stocks, then you want to keep them at the lower pace and you keep them at 18 degrees so that you don't have to transfer the fly stocks to a new container not too frequent. So these are the larval stages. In the normal block course, you would have had the opportunity to look at these larvae here and at embryos in Marcus's part, and in the adults, at the adult flies, you would also have the possibility to look at. The pupa, not many people work with the pupa. It's not so easy to work with it. So let's go on. This is how the embryo looks at the end of embryogenesis when it has turned into a little larva. This is a dark field image. Here is the head, here is the tail. This is the end of the trachea through which the larva respirates. Then you have the different segments, thoracic 1 to 3, abdominal 1 to 8, and you can see that these structures here can be very helpful when you are looking at mutants which have an influence on embryonic development, and this is what Janni Nüsslein and Eric Wischhaus were doing in the late 70s, beginning of the 80s. We will come to that in a few slides. Then here we look with phase contrast from the side. So these here are the denticle belts here you see here in front, and you can see that the back of the larva looks quite different. The hairs are more stereotyped here than they are on the belly side. So this is how the larva looks like at high magnification. Then, very important, how does the larva change into the adult during the pupal stage? During the embryonic and larval stages, there are structures called imaginal discs which develop within the larva. They have no function whatsoever in there. They are just growing. But then, during the pupal stage, many of the structures, the larval structures, are digested away, they decay, and from these imaginal discs here, the adult animal is composed. So there is an imaginal disc for each of the legs, there is an imaginal disc for the eye, and the antenna. There is an imaginal disc for the halt ears. The abdominal part here, there are no imaginal discs for it. The abdomen is formed from so-called histoblast nests, which then propagate. These cells divide and make up then later the structure of the abdomen. But almost all the rest develops from imaginal discs, and you will hear more about imaginal discs tomorrow and on Wednesday. Okay, so much about the life cycle. There would be, of course, much more to tell. We don't have the time, but this is kind of the important part you need to know at the moment. So the Drosophila genome consists of 4 chromosomes. Here you are looking at a female mitotic figure. So there are 2 X. The female has 2 X's, 2 seconds, 2 thirds, 2 fourths chromosome. In us, we have many more chromosomes, so things tend to be more complicated if you would like to do genetics with humans, but of course nobody does that for good reasons. This also means that the fly genome is much more compact than our genome, for example, and despite that, despite the fact that it is 20 times smaller, it has only about 2 times less genes than we have. So there seems to be much less space between genes in the fly than in our genome. There seems to be maybe much less junk. We don't know really what all this extra chromosomal material is doing. Maybe we will know at one point. Maybe this is part of the difference between a fly and the human, who knows. Anyway, the fact that the fly genome is so small makes it, of course, also much better suited to do genetics with it, because you don't have to follow 23 chromosomes, but you have only to follow 4 chromosomes. So this is certainly a big advantage. The fly genome was sequenced 20 years ago, and the people who were important to do all this work were Cray Venter and Jerry Rubin, and they published it in Science in the year 2000. And of course, you should imagine that this first version of the genome was not of very good quality. So the genome release 1 in 2000 was of relative poor quality. Now, 2020, 20 years later, we have the Drosophila genome release 6, and the quality of the Drosophila genome is much better. And what do I mean when I say the quality is much better? First of all, the quality of the sequence itself. It's much clearer what the actual sequence is, even for the heterochromatic part of the genome, which is not so easy to sequence because it contains many repetitive sequence elements. But it's important that in a good quality genome, the genes are the annotation of the gene is of good quality. So sequencing, of course, you all know, it's determining the basis of A, T, G, and C. That nowadays, with all these sequencing machines, has become a much different thing than when we were young in the early 80s doing our first sequences. Sequencing 1 KB was quite some work. These days, you send it out to a company, and you don't do much basically, and overnight you already have the answer. In the old days, that tended to be a little bit different. But anyway, the quality of the sequence depends also on how well the genes are annotated. So annotation means, here for example, we have the DPP gene. You will hear about much more about DPP tomorrow and on Wednesday. This is what we call the gene stretch, but you can see here DPP seems to have 4 transcripts, so it has 4 different transcripts. Here is the 5' end of the gene, here is the 3' end of the gene. You can see that the protein of DPP, although the gene is so large here, the coding region of the gene is restricted to this part here, and as far as I can tell, all the open reading frames seem to be more or less the same, independent of the transcript. Maybe Shinya can tell you more about that. I can't. But anyway, you can see that there are many other genes. Here, for example, there is a tRNA gene, which is transcribed here in the other direction, another tRNA gene here. There is a long non-coding RNA, so a gene which is not encoding a protein. A long non-coding RNA is here. Then you have many other genes on this side. If they are starting with ZG18641, for example, this is a computed gene number 18,641. DPP also has such a ZG number, but since it is a gene which has been identified by the classical geneticist, by forward genetic technology, basically, it has a gene name. DPP stands for decapentaplegic. Then you see that other gene names pop up, SLH, OAF. I don't know what they are, what they do, but gene names are abbreviated like you have it here, and it would be sometimes cumbersome to always write the full gene name, so the gene names are abbreviated to 1, 2, or 3, rarely 4 letters. If they have just been computed, then they are called CG something. So anyway, if you have a well annotated genome, then it is clear where the introns are, it's clear where the exons are, it's clear where the 5' end is, it's clear where the 3' end is, how many transcripts you have, and so on and so forth. Incredible amount of effort has gone into this, and Drosophila, together with C. elegans and yeast, are probably among the best annotated genomes that there is. Okay, so I mentioned that the genome consists of heterochromatin and euchromatin, and that the heterochromatin is localized around the centromeres. So here, these round structures here are the centromeres of the 4 chromosomes, and you can see the heterochromatin is centered around the centromere. Here, the white part is euchromatin, where you have most of the genes. Heterochromatin contains little genes. The number of genes there is small. The Y chromosome, for example, here is almost completely heterochromatic. It contains only a few genes, which are, since the male is XY, why should it have a gene, a chromosome which is essentially only heterochromatic? It contains a few genes. They are required for male fertility. Drosophila males which have no Y chromosomes are viable, but they are sterile because they lack the genes which encode proteins needed for fertility. Then you can see that the small 4th chromosome has very little euchromatin also. Nevertheless, there are some important genes also on the 4th chromosome. The 4th chromosome is only about 1% of the whole Drosophila genome. And then you can see that there are what we call chromosome arms. This is chromosome arm 2L, chromosome arm 2R, chromosome arm 3L, 3R, and the X chromosome has only 1 chromosome arm. So there are 5 big chromosome arms which contain euchromatin, and each of these arms is roughly 20% as a rule of thumb, basically. And in the euchromatin, most of the genes are localized, and euchromatin is also poor in repetitive sequences, whereas the heterochromatin contains a lot of transposons or parts of it, repetitive DNA and so on and so forth. That's why a lot of the heterochromatin has not been sequenced, only part of it. As you know, the DNA in our nuclei is not a naked molecule, but it is always associated with many proteins, and this association of proteins and DNA is called chromatin. And in a mitotic chromosome, the histones are very important to pack the long molecule of DNA into a form that it can faithfully pass on to the daughter cells, and that's how we think that it works. It's not really understood because it's not easy to determine the structure of the chromatin when it is compacted. But anyway, that's how we think about it, and as long as this model fits what we see, it's perfectly okay, even though we don't know many of the details. Here we have one mitotic chromosomes, and you should realize that in each, so here there is one DNA double helix here and one DNA double helix here, and many proteins associated with it. Here we have the centromere. If you have two arms, the X chromosome, there the centromere would be localized here at the end. The ends of the chromosome arms are also referred to as telomeres. Now, what is epigenetics? Epigenetics is something which started to become important about 20 years ago, when people realized that the N-terminal arms of the histones could be modified. So this is a diagrammatic representation of chromatin. So this is a histone octamer, and each histone octamer is made up of two histones 2A, two histones 2B, two histones 3, two histones 4. This is the histone octamer, and the DNA winds around these octamers and makes up what people called in the old days, when they did electron microscopy, it looks like beads on a string. Then you have these funny red structures here coming out of the histone octamer. And if you count these arms, there are 8 of them. So each histone here extends one of these arms. And these are the N-terminal tails of the histone proteins. And people found that these N-terminal arms of the histones can be modified. And this is a somewhat complicated picture, but the red worms here which stick out are represented here also. So we have 2B, 2A, 3, the 4th. So these are the chromosome N-terminal ends of chromosome 2A, N-terminal end histone 2B, N-terminal end 4, N-terminal end 3, N-terminal end 2A, 2B, and so these are the different N-terminal ends, and you can see this would be position 1, and then position 4 of histone 3 can be methylated. It's a lysine, so at position 4 you have it can be methylated. Then position 14 can be acetylated. Position 18 can also be acetylated. position 28 can be phosphorylated. And in this way, you can see that every arm can have a quite distinct pattern of histone modifications. And people realize now that these histone modifications are brought about by enzymes. So there are methyltransferases, but there are also demethylases. So these patterns are reversible, and they are linked somehow to cellular memory. You can imagine that each cell type has its specific epigenome, so its specific pattern of histone modifications, and these modifications, the pattern of the histone modifications, determines whether a gene which has a certain pattern of these modifications is active or inactive. And as a rule of thumb, here is the definition, a short definition of epigenetics. An epigenetic trait is a stably inherited phenotype resulting from changes in a chromosome without alteration in DNA sequence. So that's important. The DNA is not changed, but it's just the pattern of histone modifications which can change. Chromatin modifications serve as a cellular memory. They are passed on to the daughter cells, and each cell type has its own epigenome. That's in a nutshell what epigenetics is about. And as a rule of thumb, here at the top of this panel you can see that there is these beads on a string pattern. We have transcription factors, and we have here the promoter of a gene where the polymerase sits, the RNA polymerase, and you can see that the tails are modified, and as a rule of thumb, if there are methylations, trimethylation, for example, on lysine positions 9 and 27, then the chromatin acquires a heterochromatin-like structure and the gene is not active. In contrast, if the N-terminal arms of the histones are acetylated, then the histone octamers are further apart, the DNA is not as much compacted, and transcription factors have access to their binding sites and can activate RNA polymerase activity, and the gene is active. So this is a rule of thumb about how epigenetics can influence gene activity without introducing mutations on the actual genes. So, and in this way, a particular state of gene activity can be passed on to from the mother cell to the daughter cell. So that's so much for epigenetics. Now, how do we make mutations in the fly? When I mentioned to you that Morgan isolated this white-eyed fly, and they didn't have any means to induce mutations, so they were isolating spontaneous mutations. And if you have then a mutation, then at the end, starting from around 1980 on, you could try to actually clone the gene. But the step from the genetic identification to the cloning of the gene, that was hard work and took sometimes years to actually clone the corresponding gene of a mutation. So that is what we refer to, as I alluded to in the beginning, as forward genetics. So how did people induced mutations. So as I said, there are the spontaneous mutations, and there are the X-ray induced mutations. You can induce mutations by chemical means, and there is something called transposon tagging, which we will look at in the afternoon or in the second lecture. Okay, spontaneous mutations. Why did this fly here all of a sudden have white eyes? So here on this panel, also a little bit complicated, this here is the DNA, and you can see the exons, the 5 exons here of the white gene. And the white gene in the late 70s, early 80s was kind of what is kind of the topic of the day, or really important, because it allowed people to easily isolate many mutation events and study what mutations actually meant. And you can see all these triangles up here. They are all independent mutations event of different white alleles. So the white one, the first white allele isolated, had a mobile genetic element. These triangles here represent mobile genetics element. The white 1 mutation had a so-called DOC element inserted right at the 5' end of the gene, and this insertion of the mobile genetic element, the transposon, inactivated the gene, and therefore the protein of white was no longer produced. And since white protein is essential to deposit eye pigment in the fly eye, there was no deposition of eye pigment, and that's why the eyes turned white. And now you can see that there are many other white alleles. Here, white number 6, it has a P element insertion here in the 5th exon, or here white mottled 4th maybe, as a bell insertion. There is an I element, there is a rho element, there is a copia element, there is the blood element, there is the pogo element. All these are different mobile genetics elements. There are many more actually, which can insert in a gene. They move around the genome and mutate genes. And about 80% of the spontaneous mutations are due to such mobile genetic elements, the transposons. And you will see that they, through the study of these mobile genetic elements, we became actually capable of producing transgenic flies, but that will be discussed in the second lecture. So, okay, spontaneous mutations can happen. Morgan and his people isolated only spontaneous mutations. They didn't know how to induce mutation. That was then developed later by here, yes, so chemical mutagenesis. So that was much later. You can see there was a paper in 1946 here, chemical production of mutations. There was this woman here, Lotte Auerbach, together with a man called Robson. On the suggestion by Herman J. Muller, they started to experiment with mustard gas as a mutagen, and they were actually successful. Nobody would like to do a mutagenesis these days with mustard gas, of course. It was used as a chemical weapon in the First World War and had nasty effects. Auerbach and Robson also were somehow poisoned, luckily not so heavily, but anyway, they did their experiments on the roof of the institute they were working so that the wind would blow away this mustard gas. And they found that mustard gas could induce chemical mutations. Later, other chemicals were found, and one of them which we are using is called EMS, ethyl methensulfonate, and that the handling of this chemical is much easier than mustard gas. Chemical mutagenesis induces, in most cases, point mutations, and that has been used by many people. And probably the most famous EMS screen here is shown here. I have mentioned Christiane Nusswein-Volhard and Eric Wischaus. They worked as PhD student. Eric was a PhD student here at the Biocenter. Christiane Nusswein-Volhard was a postdoc in the same lab. They moved on and worked together on isolating many or most of the genes which have an effect on embryonic development. You probably remember from your lecture, which Marcus gave to you in your second year, names like Wingless, Odd-Skip, Fujiteratsu, and so on and so forth. So they looked at mutations which would change here the wild type pattern into a pattern like this, the pair rule, or here triple, knirps. They are the so-called gap genes. So this was an EMS screen, and here on the next slide I have shown you the crossing scheme of this screen, and at the latest on Friday you should understand how this crossing scheme worked. We will look at that on Friday afternoon, and I will explain to you how the screens they did worked. At the end of the day, what they had done is they had screened 27,000 mutants, basically, 580 of which had a dramatic embryonic phenotype, and these 580 could be classified into 139 complementation group. So at the end of this week, you should be able to understand what all this information is, what complementation is, how this screen was done in the first place, and we will move on right now because we want to know how they could actually do this screen at all. For example, what is TM3? We will come to that in a minute. It's a balancer chromosome, and we will talk about balancer chromosomes soon. Okay, we had spontaneous mutations, we had chemical mutagenesis, and then come now the X-ray induced alleles. So the man who developed this technology was Herman J. Muller, the postdoc in the Morgan lab, beginning of the 1910s, and he published an article called Artificial Transmutation of the Gene, in which he described that by using X-rays he could increase the mutagenesis frequency by a factor of 100, and that was of course spectacular. So what do you get when you do X-ray mutagenesis? X-rays break the chromosomes and they are put anew together. So you can make deletions or inversions, duplications, translocations, and transpositions. And that is depicted here on this slide. So a deletion, there is a break here and a break here, and you lose the DNA. That's why the segment DE is lost here. Or you duplicate the D part. There are inversions that you have a break here and a break here, and this part here reinserts into the chromosome, but in the opposite orientation. Depending on whether the centromere is included, one distinguishes between pericentric and paracentric inversions. Then there are translocations where genetic material is exchanged between two non-homologous chromosomes. So in this case here you have the red chromosome and the white chromosome, and after the translocation has occurred, you have now two mixed chromosomes consisting here of white genetic material and red genetic material or red genetic material and white genetic material. Then there is also the possibility that only part of one chromosome moves on to another chromosome. This is called transposition. How did people find out that these chromosomes actually look like I'm showing you here, that there were actually something like deletions, duplications? I will show you in a minute. For the moment, we will concentrate on the inversions. As I said, if you make an inversion, you have two breaks here, and the DNA in between here is flipped around, and you have here the normal chromosome, 1, 2, 3, 4, 5, 6, 7, 8, 9, and the inverted chromosome, 1, 2, 3, 6, 5, 4, 7, 8, 9. So that doesn't seem to be big problem unless the breakpoints here are in an important gene. Then you have a mutation. But apart from that, an inversion could be a viable chromosome. And in fact, people have found in wild-type stocks that there are such inversions around which do not have an obvious phenotypic effect on the appearance of the fly. But in the cell, this has an effect because in Drosophila, the homologous chromosomes want to pair with each other. And if you have an inversion, when these chromosomes here want to pair, then you have a problem because this region 4, 5, 6 is no longer in the same order. And you would imagine one way to accustom this inversion would be to make such a loop. And people actually could identify that there are such loops upon induction of an inversion. For that, they needed to use the so-called giant salivary gland chromosome. So during larval development, some cells start to polytenize, meaning that there are endocycles, so the DNA replicates, and instead now having a mitosis, there is another replication step and another replication step and another replication step, and this can go on for a while until you have about 1,000 copies of DNA in the same nucleus, and all these chromosomes want to pair with each other and they align. So these chromosomes you would have looked at in today's practicum. Unfortunately, you cannot do it. Here, by the way, is the small fourth chromosome. This is the chromocenter. The chromocenter is under-replicated, meaning that it doesn't go through all these endocycles. So there is much less heterochromatic DNA, and it all coalesces here in the chromocenter. So what you are seeing here are the euchromatic chromosome arms, and each of them has a distinct pattern. So there are bands and interbands. Interbands is the whitish looking, and the bands are the dark parts, and every chromosome has exactly the same pattern of bands and interbands. So 2R in one fly looks exactly the same as 2R in another fly. And by studying these patterns, they could identify the structural changes which X-ray induced mutations would have done. So here, just as a comparison, the mitotic chromosomes I was showing you here in relation to the size of the salivary gland chromosomes, you can see the salivary glands are way bigger than mitotic chromosomes. And by studying here the pattern of the bands and interbands, people could identify the breakpoints of the X-ray induced mutations. Here there is the loop which I propose to you an inversion should have. So here is this chromosome part here, and you can see telomere, telomere, and then the inverted part. This is the loop which forms. And by now reading the pattern of bands and interbands, you can determine where on the chromosome the breakpoint has occurred. Of course, not by very high resolution, but you get an idea where the breakpoints have occurred. Okay, so this is how inversions look like. And these inversions, they are now very important to understand how balancer chromosomes work. People found when they were inducing these inversions that in meiotic recombination experiments, that chromosomes containing inversions didn't behave in the way people expected, meaning that the recombination frequencies which were measured between a gene located here and somewhere in the loop here were dramatically different from what it was on a normal chromosome, because, and the reason for that was, also the recombination frequency was more or less zero, didn't recombine anymore, and people found that these inversions were hindering recombinations or were leading to unviable recombination products. And this is a very important feature of balancer chromosomes. They seem to suppress meiotic recombination. And I have prepared some slides which should kind of lead you to why balancer chromosomes are such an important tool in fly genetics. Balancer chromosomes, very important. You should understand these couple of slides to understand how balancer chromosomes work. So I mentioned to you that Christiane Nüstern-Volhard and Eric Wieschaus isolated many lethal mutations, homozygous lethal mutations, which didn't have a phenotype as heterozygous. So if there is one lethal copy here, one gene with a lethal mutation and a wild type copy, this fly here doesn't have a phenotype. So how should you select for it? So let's assume that you are crossing two heterozygous flies, and Mendel says that there are three classes: the homozygous wild type, the homozygous lethal, and they die, and again the heterozygous lethal individuals. But again, you cannot identify them. These here die because they are embryonic lethal. So how should you ever be able to maintain a stock containing this lethal mutation if you cannot select for it? If you think of this crossing scheme here, you will realize that at the end you have lost your lethal mutation and you can never work with it again. So the solution for this problem is balancer chromosomes. Why? I will show you in a second. So that's one thing. You need balancer chromosomes to maintain embryonic lethal mutations, for example. Then another problem is that chromosomes may not only contain one important component, but several. So in our case here, I have drawn a lethal mutation here and a marker associated with it on the same chromosome. And during meiosis, these two markers can separate from each other, and of course you don't want that. The recombination frequency depends on how close these two are. If they are very close, you are not very likely to have a recombination event, but if they are far apart, the recombination frequency will be much higher. Anyway, the idea would be that you keep these two genetic markers together in this, so like this here. So if you cross these flies with each other, you can get back the parental chromosomes, this one and this one, but you can also get the recombinants. And as I said, you are not interested to lose the linkage of the lethal and the A marker. Also here, I already mentioned that inversions are suppressing the recombination events, so balancer chromosomes come in handy to keep the integrity of chromosomes as you would like to keep them. So how do the balancer chromosomes do that? How do they prevent recombination? So inversions suppress meiotic recombinations, and that is what balancer chromosomes are about. They are consisting of many inversions. Here is a short explanation how these inversions suppress it. So here we have two chromosomes. One contains an inversion, the other not. And now we have here a recombination event within the inversion. So you come here and then you move over and you continue here, or you come in here, you move over and you continue here. At the end of the day, you will have a chromosome which has no centromeres and a chromosome which has two centromeres. And you can imagine when the chromosomes have to be distributed to the daughter cells, this is a major problem. This chromosome here you will lose, and this chromosome will be broken apart very likely, and you will lose it. In addition, here you have lost part of the genetic information, so there is a deletion, and also on the other one you have lost genetic material. So these chromosomes are dominant lethal. Even if they would be transferred to the daughter cell, they would be lethal. And I mentioned that there are the paracentric inversions and the pericentric inversions, but independent on the type of inversion, the resulting recombination products upon recombination within the inversion leads to chromosomes which are not viable, because either they have no centromeres or they have two centromeres, or in the situation over here, you have again large duplications and large deletions, and these are dominant lethal. Okay, so that's how things work in balancer chromosomes. That's how they suppress meiotic recombination. Meiotic recombination still occurs, but the recombination products are dominant lethal, and that's how they will never show up in the next generation. I show you here now a number of examples for balancer chromosomes. Here, this is a balancer for the first chromosome, so the X chromosome. It's called first chromosome with multiple inversions, number 7. This already tells you that there are numerous balancer chromosomes for the X, and here are listed all the markers which are on it. There is yellow, there is skewed, there is white apricot, there is singed, there is vermilion, there is garnet, and there is bar. And as you can see, bar is written with a capital letter, and this means it is a dominant marker. These are all written with a minuscule letter, they are small letter, they are recessive. But bar is dominant, and this is the phenotype. This is a wild type eye, and bar reduces the size of the eye dramatically. And this is, when you see this phenotype here in a cross which contains FM7, you know this fly here contains the FM7 chromosome. And if we now look up at the X chromosome, here wild type, the numbering from 1 to 20, now we go look and see that the FM7 chromosome 20, which normally is over here, is now over here. But then it continues with 15, 16, 17, 18, 19, and then there is another inversion, 14, 13, 12, 11, 4, 5, 6, 7, 8, 9, 10, and then over here is 2, 3. So you can see that all these inversions completely have changed in the way in which the chromosome is put together. And in this way, the FM7 balancer chromosome for the X is suppressing the recombination, and you can use it as a balancer chromosome to maintain lethal mutations on the X or to maintain different markers which have been put together on the X chromosome. Here, this is how the geneticists write the breakpoint. So you can see 1 here is this number 1, and then it continues with 20. That's this small piece 20, then 15 to 20. So this is the cytology, which basically describes the order of the inversions and other rearrangements event on the X chromosome. And to determine these breakpoints, here, here, and so on, people were looking at salivary gland chromosomes to determine these breakpoints. Of course, as I said, this is not very precise, but sufficiently precise for the geneticist of the time. There are balancers for the X chromosome, for the second chromosome, and the third chromosome. This is the balancer for the second chromosome. It contains, again, many inversions, as you can see by comparing the wild type from the curly. Curly O is the name of this balancer chromosome, and you can see it contains a number of markers: dumpy, prune, cinnabar, curly, and here curly is the dominant marker. It's spelled with a capital C, and curly causes the fly wings to be curled up like here shown in the picture. Then the third chromosome marker, TM3. Here the dominant marker is serrate. Not clear to me why it's not shown here. And it also has stubble. And again, you can see contains many inversions. This is the wild type third chromosome going from 61 to 100. And this is the rearranged chromosome. And this is something we have to mention in a minute. So, but here, okay, this would be the dominant marker on this TM3 stubble. These are the hairs in a wild type, the bristles in a wild type fly, and you can see that the stubble, the dominant stubble, causes the morphology of these bristles to change dramatically. They look much shorter than they do in the wild type. So let's go back again. The TM3 balancer chromosome is not a good balancer if your gene of interest is localized in this part here of the third chromosome, because there are no inversions in that region. So if you know that your gene is localized in this part of the third chromosome, you should not use TM3 as a balancer, because then you are very likely to lose your mutation. For that, you would rather use the TM6C balancer. It has, again, stubble as a dominant marker, and ebony is a recessive marker here. And you can see now, if you look at the tip here of 3L, it starts with the telomere 61 and then not 62, but the inversions continues here. So TM6C would then be the perfect balancer for you if your gene would be localized in this area here. Here there is another marker mentioned. It's called TABI. TABI has a phenotype which can also be identified in larva and pupa. That's why we sometimes use it to select larvae which are not TABI. So here, this is a wild-type larva. You can see it's long and thin, and the TABI are short and chubby. So you can easily distinguish these two. You would have actually done that if we would have done the practical part of the course. You would have distinguished between TABI and non-TABI larvae. The difference is also apparent in the pupa. The wild-type pupa is clearly longer than the heterozygous TABI pupa. So this is a marker which can be used not only in the adult but also in larval and pupal stages. There are not so many markers like that. Okay, we skip the MKRS and go to the important characteristic of balancer chromosome. They must contain at least one dominant marker that we can follow them. The autosomal balancers for the second and third, they are homozygous lethal. The situation for the X chromosomal balancer is a little bit different. This will be part of the genetic questions. We will discuss it there, why X chromosomal balancer chromosomes cannot be homozygous lethal, but they are for the second and third chromosome. We don't need actually balancer chromosomes for the fourth chromosome. There is, because it's largely heterochromatic, there is no meiotic recombination on the fourth. Balancer chromosomes contain multiple inversions which suppress the formation of viable recombination products. And this is now a crossing scheme where we have now our lethal mutation over a balancer. We cross males and females together. And if we follow Mendel's rules, there are three types of progeny: homozygous balancer, homozygous lethal. These guys here die. If we have this mutation on the second or third, we will never see them. Also the homozygous lethal, we will of course not see them. The only progeny which survive is lethal over balancer. So they are exactly the same as the parental genotype. And if you cross these again, the only which survive are lethal over balancer. So in this way, you can maintain the lethal mutation for many, many, many generations, decades. Basically, you can maintain your lethal mutation, and that's why balancers are so important. You never have to select for the lethal mutation. It is always there. Later with the when people found that GFP can be introduced into flies and many other animals, of course, so-called green and blue balancers were created. The blue balancers have a copy of the lac-C gene, which you probably know from your microbiology block course. You can stain, if you have lac-C in there, you can add a substrate and the tissue will turn blue. Or if you have, this is for fixed material. If you want to work on live material, GFP, you can see it under the fluorescence microscope. And in this way, if you have, for example, a GFP marker on a balancer chromosome, you can also select your animal of choice under a fluorescent microscope. So here, for example, we have the green balancer. You cross these flies with each other, and everything which doesn't have the balancer is not green. So in embryos, let's assume this is embryonic lethal. If you then check which embryos are green, you know they are not the homozygous, but you are of course interested in the homozygous animals, and you can select them by absence of the green signal. So the balancers, with the help of GFP, become also useful earlier than just the adult stages. Balancer chromosomes in other model organisms have been recently developed because people realize they are important or can be of help. Mice, of course, can be genotyped and have a much longer life cycle, so maybe not so of big use as they are in fly, but here there is a case for chromosome 11 of fly. There is these two genes, TRP53 and Wnt3, which normally have a recombination frequency of 24%. Then, by using the Cree recombination system, inversion was introduced, and now the recombination frequency, if you have this inversion there between Wnt is now on the other side and TRP53 is now on this side, the recombination frequency drops to 0%. So these inversions work exactly in the same way as they do in the fly, and they use a dominant marker. Here we have balancer and wild type. The balancer mouse has less pigmented fur than the wild type. So basically, these mouse balancers are designed according to the fly model. Okay, so briefly let's go through the classes of mutations. Herman J. Muller realized soon that not every mutation is a loss of function mutation. So normally you think when you introduce a mutation you destroy the gene, but that doesn't have to be the case all the time. So loss of function mutations, you distinguish between hypomorphic and amorphic. Hypomorphic meaning that the gene retains some activity, hasn't lost all. Amorphic meaning it's basically a null mutation. The gene has lost all its activity. Then there are also so-called gain-of-function mutations. So that means these can be mutations where more of a gene product are produced. These are hypermorphs. And there can also be situations where you have ectopic expression, where you produce the gene product in a tissue where it normally is not present. And this can also have dramatic effects on the morphology of the fly. Then another important class are the dominant negatives. These are mutations which are in a way more severe than an amorphic mutation because they are poisoning the complexes it normally takes part in, also in protein complexes, and renders them inactive. And I have a figure where we skip this. These are all the names you should know, which I also mentioned. So the dominant negatives here, they are a special kind of mutation. So these are mutations which still produce the protein, but they produce a gene, a protein which is not really active anymore, but when present in a protein complex can cause this complex to become inactive. So let's look at the most simplest case of a homodimer complex. Here, this is a heterozygous situation. We have a gene, a small deletion which removes the gene, and a wild type copy, and this wild type copy produces a protein, and when a dimer is formed here, this basically works okay, and as long as one copy of the gene is sufficient, which is the case for most of the genes, or in the fly, then you will not realize that actually one copy here is missing. Now in the situation of the antimorph, dominant negative, you have the antimorphic mutation over a wild type. Two types of proteins are formed, the red from the wild type and the blue from the antimorphic gene. And you realize there are now several types of dimer complexes which can be formed. There is the homozygous red, there is the homozygous blue, and the heterodimer between the red and the blue. And if now blue is inactive, only one of the three complexes is actually active. And in this way, the antimorph can be more severe than just the plain loss of function. Now, how do you demonstrate that your mutation is of antimorphic nature? If you increase by a duplication or other means the concentration of the wild type protein, then you increase the number of wild type homodimers, and you should suppress the phenotype induced by the antimorphic mutation. It's a little bit complicated, but I'm sure if you think of it, you will see how this antimorph works. Then we briefly discuss the gain of function and loss of function here for few homeotic gene mutations. And normally the fly has two wings. So we are looking now at the ultrabithorax UBX gene and what the consequence of loss of function in UBX is. UBX is mainly expressed here in the thoracic segment T3 and to a much lesser extent in the thoracic segment T2. Now, if you reduce the amount of ultrabithorax, you get this very dramatic four-winged fly. And if you look at the protein levels in thoracic segment 2 and 3, you will see that T2 has, in T3 there is much less, and the third thoracic segment, which normally makes these little hole tears here, is changed into a copy of T2. T2 is where the wings are attached. So this, we would have to go into homeotic gene genetics, why this is like that. We don't have the time for that, but this is a typical example of a UBX loss of function phenotype. What happens now in the alternate situation where we cause more UBX in T2, keep the levels of these UBX in T3, but increase these levels here to a higher level. I showed you before, if you lose UBX in this area, you transform them to T2, and now if we increase the UBX level in T2, we transform the wings into halt ears. So this is a fly which has lost. Mutation is actually called contrabithorax, and in an extreme case you can see that now the wings are turned into halt ears here. So this is a dramatic situation of a gain-of-function mutation where UBX, levels of UBX are changed, leading to very clear-cut phenotypes. Okay, then Antennapedia. Also here you might have heard about it. This is the depiction of a Drosophila head. Here are the antennae. This is the arista here, and this is also part of the antenna. The arista is part of the antenna. Here the fly eyes, here is the mouth of the fly, and now normally Antennapedia is not expressed in the I-antennal imaginal disc. So no expression, but in the, for example, in the Antennapedia 73b mutation, all of a sudden Antennapedia is expressed in the I-antennal disc. We say Antennapedia is ectopically expressed because it's now present in cells where it normally is not, here, and as a consequence the antennae are turned into legs, and it's a T2 leg because Antennapedia determines the identity of the second thoracic segment. So this is an example of an ectopic gain-of-function mutation. Then if you have a mutation in a gene or several mutations in a gene, they do not all need to have the same phenotype. Here are examples for Fujitaratsu, one of the parallel genes. The wild type you know by now. Here it has 3 thoracic and 8 abdominal denticle belts in the wild type. In the null allele of Fujitaratsu you can see that there are only 4 of these denticle belts left. And now there are hypomorphic mutations where you have different parts of this wild type pattern coming back. You can see here part of this denticle belt comes back. And in this way you have a whole phenotypic series of different mutations giving rise to different phenotypes. So this is called a phenotypic series of alleles of the same gene leading to different phenotypes. Okay, that's it for the moment. Took much longer than I thought. That's normal. But we will anyway have a break now because everybody is exhausted, I guess. Yes, so let's say we meet 20 past 11 to continue for the reverse genetic part of these Monday lectures. So a lot of new information. I hope you got some of it, and as I said, there are opportunities to ask questions, and see you in 25 minutes, 20 past 11.